السلام عليكم. محاضرة Antimicrobials 1. نبلش. What are antimicrobials? So these are the medications that we use to treat infections that are caused by microorganisms, antimicrobials. We usually use them when these microorganisms overcome our defenses, and these are aimed not only to treat bacterial disease but في عندنا antivirals, antifungals, antiprotozoals and antihelminthes drugs. هدول الميديكيشنز غيروا كثير من يعني هم والفاكسينز صراحة غيروا كثير من يعني الحياة، يعني they have changed dramatically, changed the game. قبل يعني 100 عام أو أكثر ما كان فيه، يعني قبل الأربعينات ما كان فيه أي antimicrobials. You had nothing. Somebody had infections, they had to overcome it on their own. They didn't, خلاص they will expire. قبل هيك we didn't have any vaccines. زي ما انتم شايفين هلا احنا in the current pandemic we have few treatment options and no vaccine. فكل الدنيا وقفت، كل الدنيا it was affected severely. So we can see how important it is to have antimicrobials, antivirals, antifungals, antibacterial drugs. هلا مشكلة عندنا that هدول الأدوية لهم ثمن. What do we pay for using these drugs? أولا شيء هو side effects. There are side effects that sometimes can be serious. Some of them are even permanent. عدا عن هيك احنا هلا عندنا ما إننا تقريبا 80 سنة نستخدم هاي الأدوية أو أقل. هلا احنا عندنا كثير we are suffering from and facing a crisis of antimicrobial resistance, and we know how that happens with all the horizontal gene transfer that the bacteria has. So it is عشان هيك لازم احنا نعرف how, when and why we can use these drugs and to understand all these consequences. فantibiotics عادة كلمة antibiotics مش antimicrobial عادة it denotes something that has been naturally occurring microbial product. عادة penicillin إذا سمعتوا عن penicillin, it comes from a penicillium fungus. They grew this fungus and they saw that this fungus kills the bacteria around it. No bacteria can grow around it. أو يعني a lot of bacteria was not able to grow around it. درسوا شو في المشكلة، وجدوا إنه it has a naturally occurring substance that kills bacteria by destroying its cell membrane. فاستخدموه، صار في عنا drug اللي هو أخذوها they harvested this and they made it and then we had the first antibiotic which is penicillin. هلا الكيموثيرابيوتكس are chemical agents. They are synthetic and مثلا راح نحكي عنهم مثل drugs, sulfonamides, amidazoles, nitrofurans، كلهم هدول راح نحكي عنهم quinolones. So هدول some antibiotics are manufactured synthetic, some are even we chemically modify a naturally occurring substance. فبصير هذا semi-synthetic. There are a lot of penicillins that have modification of their beta-lactam or their structure can produce different pharmaco effects which can improve and solve certain problems. So بما إنه في overlap between synthesis and naturally occurring substances فبنستخدم إحنا كلمة antimicrobials. So an antimicrobial substances are too toxic to be used. Where are we? Okay. So this is إحنا بما إنه في عنا overlap between, so we are used to describe these are used to treat systemic infections. From antimicrobial substances that are too toxic to be used systemically can only be used as typical drugs. هدول مثلا نسميه مثلا antiseptics. يعني إذا بقدر أستخدم an antiseptic systemically صار هذا antibiotic، صار antimicrobial. That's what this means. So antiseptics هم disinfectants is the answer. فنطلع مثلا على المشكلة اللي احنا بنواجهها حاليا. So this is the number of systemic antimicrobials. دول الانتيميكروبيالز اللي قاعدين بنستخدمهم and they have went through from phase 1 to phase 4 or phase 3 of clinical trials and they passed and they work and everything is fine. فاحنا عندنا العدد قاعد بقل. كان عندنا 15 بالثمانينات، طلع 15، بعدين طلع 10، بعدين now we have less than 10. Now we have in the last period less than 5. So we have fewer antimicrobials, new antimicrobials being released. وعندنا resistance قاعدة بتزيد بسرعة. المشكلة إنه resistance is happening much faster than the rate of our introducing of new antimicrobials. We are losing this battle. ما بنقدر إحنا نطلع new antimicrobials fast enough to combat antimicrobial resistance. هذا بطلع هذا this is one of the pages يعني صفحة كتير حلوة طلع عليها is called the resistance map. بتشوف resistance for certain microorganisms against certain antimicrobials. فبتشوف مثلا الامينو penicillins for مثلا Klebsiella. عفوا this is for E. coli and you can see penicillin يعني it's a gram negative. البنسلين تقريبا 50% أو أعلى it is resistant. في أنت مثلا carbapenems, this is one of the last options that we have against resistant pathogens that are E. coli. It has no resistance. بعدين صار عندنا uptick بداية آخر 2011. So now we are permanently in this uptick. Carbapenem resistant Enterobacteriaceae is a very big problem. Amoxiclav. So when you combine amoxicillin with clavulanic acid رح نحكي عنه. So the resistance is less, but it's still on the rise. And you can see مثلا for Klebsiella is something similar. مثلا carbapenems قبل طلعت بال 2004 بلش and it's very high for Klebsiella. 10% carbapenem resistant. And it's similar for the other substances. هلا في نقطة طلع عليه هنا إنه things can come down. So نطلع على amoxiclav it has reached about 30%. نزلت 20 حتى تحت 20. So things can come down. How do antimicrobial resistance come down? Well, if you stop the use, إذا ذكروا محاضرات الجينetics, if you stop the use of a certain antimicrobial لفترة معينة، هذا antimicrobial resistant gene وين موجود؟ on a plasmid. So if the plasmid is not being used، هاي الفلاش درايف أنا ما بستخدمها. So less and less people have it. أنا ما استخدمتها. I was never come in contact. ما بستخدمها. So you can actually reduce by limiting the use of antimicrobials. And this is the point of antimicrobial stewardship in how to control the use of antimicrobials. إيش بنستخدم شو and to control the rate of their use. هلا the prime and most vital and important concept اللي هو بالantimicrobial therapy اللي هو selective toxicity. تذكروا حكينا إنه I want to find a target بالمايكرو organism أنا ما عندي منه هذا التارجت. فأنا ما عندي cell wall. So if I target the cell wall I will not harm my own cell. If I target their ribosomes, they're different than my ribosomes, I will not harm myself. If I target their DNA, they have prokaryotic DNA, but this I still have DNA. So anti-DNA antimicrobials ممكن يكونوا فيهم عندهم يعني أصعب unless I target an enzyme that replicates DNA that is different from my enzyme. وهكذا. So that is called selective toxicity, is to exploit these differences between the metabolism and structure of the microorganism as opposed to our own cells. مثلا the penicillins والcephalosporins they are effective and selective because they stop the bacteria from producing peptidoglycan. احنا ما عندنا peptidoglycan. فالmechanism of action of antibacterials and antifungal drugs. So they are on three main targets. وشو هم؟ الخلية عبارة عن DNA، بروتينات، ribosomes and cell wall. So I either target the cell wall, I either target protein synthesis, or I target nucleic acid synthesis. That's all I have. So we will see إنه رح نحكي عنهم دول كلهم in the next 3 lectures. So um, antibacterial activity مثلا inhibition of transpeptidization تذكروا حكيناها peptidoglycan formation. هذول penicillins, cephalosporins, imipenems, aztreonam, vancomycin. other step راح نورجيكم صورة cycloserine basic tracing also works. بالفنجس راح يكون هو الكابسول فنجن. في عندي 50S ribosomal subunits and 30S ribosomal subunit. بال 50S so عادة نحكي على مثلا 30S we will talk about aminoglycosides and tetracyclines and then chloramphenicol, clindamycin and erythromycin. Inhibition of nucleic acid sulfur drugs, quinolones, rifampins دول راح نحكي عنهم. other membrane functions polymyxins, daptomycin, amphotericin in the antifungals. كل هذا راح نحكي عنه إن شاء الله. هلا this table now we want to start looking at certain patterns عشان تساعدنا هاي الأنماط تساعدنا to keep track of all these antimicrobials and how we can ولو بسرعة لو كان عندنا مثلا certain rules that we can use that can help us maintain يعني a good mind map about these drugs. فإذا بطلع أنا مثلا the penicillins, the cephalosporins and the glycopeptides, the vancomycin اللي هم and the other beta lactams. So these have a site of action هون. هلا أنا مثلا if I take them against staphylococci هم gram positives, streptococci gram positive, these are the dominant gram positives راح استخدمهم or the most common. بعدين عندنا enterobacteria أو enterobacteria اللي هم enteric gram negatives. في عندي then the three special cases هم أنا بستخدم عليهم البكتيريا. So the gram positives ممثلين بهدول gram negatives بالenteric bacteria and Pseudomonas, Mycobacteria and anaerobes are the special cases. Pseudomonas ليش special case? It's a biofilm former, it's a very strong biofilm former, has a lot of antimicrobial resistance. Mycobacteria ليش حكينا ذكر lipid rich cell wall. Anaerobes why? Because they have a different respiration mechanisms. So بحط أنا I plant my three site of action cell wall, ribosome, RNA DNA synthesis. And then I start to see the patterns. هون بشوف شو أنا بقدر أطلع. أول إشي بشوف إنه مثلا my cell wall synthesis drugs don't work on all the special cases الثلاثة. They have variable activity على Pseudomonas. Actually, they don't have any activity on Pseudomonas except for special drug penicillin, a special drug cephalosporin, and a special drug other. So there's only one class for each that works, but others don't work. Mycobacteria they don't work. Anaerobes they do work, but on variable for poor activity طلع هون مثلا against certain groups. So it's a little bit difficult. بطلع عليهم كمان I have a lot of resistance. R يعني resistance, V يعني variable. Plus works, minus doesn't work. So a lot of work, it does work on the staphylococci and the streptococci. Penicillins have a variable activity on the enterobacteria, but they do work. The others, cephalosporins and the gram other works. So that's for the first mechanism. هلا second mechanism الريبوسومز ايش الاشياء الادويه بتشتغل على الريبوسومز من aminoglycosides, tetracyclines and chloramphenicol, erythromycin and the others. بتطلع all of them have a great activity on the gram positives ما عدا aminoglycosides. They don't work on some of the streptococci, but they have a good activity. هلا بالعكس تقلب الحالة إنه a lot of them don't work on the gram negatives except for these the ones مثلا اللي هم tetracyclines, aminoglycosides, and then we go to Pseudomonas, nothing works on Mycobacteria, only have these rifamycins and some one or a few aminoglycosides, and the other special case for the anaerobes we have the macrolides are a good example here. بروح على DNA RNA mechanisms. You can see they again it's very hard to use on Pseudomonas, very hard to use on Mycobacteria except for the quinolones. And then here where anaerobes nitrofurands are they have good activity against anaerobes. فاطلع مثلا anaerobes شو اشتغل عندي أنا I have aminoglycosides عفوا microlides and some of the cell wall affecting drugs. Mycobacteria I have only rifamycins and quinolones that work. Pseudomonas some of the cell wall drugs work and some aminoglycosides. And enterobacteria كنت بشتغل مثلا quinolones, aminoglycosides, chloramphenicol, and the cephalosporins and other beta-lactams. gram-positive شو بشتغل عليهم? A lot of things work on them. So that means gram-positives are the least resistant. بس في variability بالresistance بالgram-positives. Almost everything works. Nitrofurands really are not prescribed for gram-positives, but we have some resistance patterns. So هاي يعني يعني زي بتحيط إذا يكون عندك مثلا quick understanding of the activity against antimicrobials and antibacterial agents. So نطلع احنا مثلا من الاشياء نشوف نعرفها كمان انه we have a lot of antibacterial drugs. Then we do antiviral drugs because we have a lot of targets on the bacteria. The bacteria is self-replicating. It has ribosomes, it has DNA, it has RNA, it has the cell wall, has everything. The virus has a small amount of DNA, small amount of RNA, a viral capsid or protein. So it's less targets and the virus بيستخدم my own machinery to replicate. So it's very hard to selectively kill the virus. And here's what we say is إنه بنستخدم normal human function to replicate and distinction is much hard to achieve. So شو معنى broad spectrum versus narrow spectrum؟ narrow spectrum مثلا بيشتغل على مثلا نوع واحد من البكتيريا gram positive staphylococcus مثلا. broad spectrum بيشتغل على gram positives gram negatives anaerobes شو اسمه pseudomonas بيشتغل على mycobacteria. So we have works a lot against a lot of things. That's a broad spectrum antimicrobials. هلا narrow spectrums are actually active against one or very few types. So that's the spectrum we are talking about. Spectrum اللي هو اللي هون هذا spectrum. So here حكينا معنى bactericidal versus bacteriostatic. So it's important to see this. So راح يكون we know this, the salient features of behavioral bacteriostatic drugs اللي هم they can grow again. لما تشيل الدرج حكينا the bacteria will grow back and it depends on your own systems to clear the bacteria. انت بس تعطي your own system يعني النفس زي ما حكينا so it can continue so it can catch up and destroy these bugs. هلا bactericidal drugs are particularly useful. Icidals إذا مثلا إذا عندك واحد he has an immediate life-threatening infection ما بتقدر تستنى his system to start working فبتعطيه بتستخدم يكون اتجاهك أكثر للبكتيريcidal drugs. Patients ما عندهم أي شو اسمه immune system فما بتقدر تستخدم his own immune system to work. So we are going to focus on bactericidal drug something that can cause immediate destruction مثلا endocarditis. So phagocytosis is limited by these vegetation with the biofilm or slime or whatever, so bacteriostatic will not be working. You will need a bactericidal drug. So that's the figure. This is the growth pattern. When you add a drug, bacteriostatic بعمل لك هنا bacteriostatic drug removed برجع لgrowth. If there's no drug added راح يضل growth شغال. If you add a bactericidal, so you can start seeing the death curve right away. So inhibitors of the bacterial cell wall هل راح نبلش نحكي عنهم. As most bacteria possess a rigid cell wall that is lacking in mammalian cells, this structure again is a prime example for or a prime target for selective toxicity as it is we don't have a gram ما عندنا cell wall احنا. So however, the bacterial cell wall can also prevent access of agents that would otherwise be effective. هذا معناته بدي راح يغلبني إن أدخل certain large molecules to inside the cell. So inhibitors للبكتيريا cell wall work mainly on the peptidoglycan اللي حكينا هاي موجودة بالgram negatives and the gram positives and both of them cannot live without it. So it is a good target for both. فنطلع مثلا peptidoglycan layer حكينا this is an active dynamic structure زي skin, it's always being modified, deposited, and the old is being removed. So if you stop its synthesis خاصة على newly formed cells, that will be destructive to them. That will be bactericidal to them. هلا for old cells this will be bacteriostatic, بس عادة old cells are in a few amount هذا مقارن مع الكبار، بما انهم انه البكتيريا خلايا متجددة بسرعة. فبتشوف انه your beta lactams are the dominant هو بيستخدم على cross linking peptidoglycan transpeptidation, so that's where beta lactams work. Glycopeptides مثلا الفانكومايسين بيشتغل مثلا على transfer from the lipid carrier to peptidoglycan. Basic tracing works on the dephosphorylation of the lipids. Phosphomycin تحويل sugars بيشتغل عليهم. and cycloserine على الامينو acids، فعندنا sugar amino acids, so these work on each. These are all the mechanisms of the peptidoglycan layer formation. بتذكروا هذه من المحاضرة الأولى, so just as a reminder I put it here. فأول شيء نحكي عن البيتا لاكتام agents اللي بنضمن لهم هم البنسلينز والسيفالوسبورينز, يعني probably the most prescribed antimicrobials خاصة بالعيادات. Penicillins and other beta and cephalosporins, they contain this beta lactam ring in their structure. هذا البيتا لاكتام they bind to proteins that are situated in the junction between the cell wall and the cell membrane. هذول اسمهم بروتينات اسمهم penicillin binding proteins. It is a bacterial protein that is present there at this junction, and they are involved in the هم بيعملوا cross linking للبيبتيدوغلايكان. هم بيركبوا هالبيبتايدز مع بعض, هم بيركبوا هالكروس لينكينج بالباك بون تبع البيبتيدوغلايكان. So if you put this beta lactam on it, it will bind the penicillin binding protein and prevent it from continuously cross-linking the peptidoglycan. فما راح تصير زي ما قلت تخيط هالحيط, فهي بتوقف هذه اللي هي الخياطة اللي هون. So this chain will not start to be formed. So these enzymes هم بسموهم هلا في شيء enzyme اسمهم beta lactamases which destroy this beta lactam agent and allow the penicillin binding protein to work. حكينا هذول enzymes موجودين, ممكن some of them are present in the space, in the periplasmic space in these gram negatives. These have gram negatives enteric bacilli مثلا have particular diverse beta lactamases. They are not all the same. They all break down different types of penicillins and cephalosporins. And the most prevalent اللي هو TEM beta lactamase, TEM1 and 2, and that is the most dominant form of beta lactamases. هلا beta lactamases حكينا present a lot of gram negatives and produce it, and it can activate a lot of types. هلا في شيء جديد صار يطلع اسمه extended spectrum beta lactamase which has more activity on a lot more penicillins and cephalosporins, وهذا صار كثير مشكلة كبيرة عندنا. It is one of the dominant or the problematic enzymes for bacteria to have and causes a lot of infections because it limits a lot of our use for antimicrobial choice for treatment. Penicillins, the original نبلش نحكي عن البنسلينز. أول penicillin هو original penicillin سموه أو يعني كلمة OG مثلا, this is my old school, يعني the old school penicillin is the penicillin G. It is often called simply penicillin. إلى الآن لازلنا نستخدمه. It still has activity against Staphylococci, Streptococci, Neisseria, Spirochetes مثلا مثل Borrelia, Treponema, and other organisms. Penicillins, they act by inhibiting transpeptidation حكينا, and the enzymes that catalyze the final cross-linking in the synthesis of the PG layer. حكينا resistance on these is on the rise, especially against penicillin G. Added to that, there are a lot of types of bacteria that some Staphylococci and مثلا especially pneumococci have less susceptibility to penicillins are now being encountered. فبما انه هذا البنسلين طلع لنا البنسلين G was يعني الريفوليوشن. التقيت بدكتور كثير كبير بالعمر، والده كان كمان طبيب، والده كان طبيب بالثلاثينات، فكان يحكي لنا عن والده. يعني والده عاصر الفترة قبل الانتي مايكروبيالز وبعد الانتي مايكروبيالز. بالنسبة لهم الانتي مايكروبيالز كانوا سحر. يعني كان هالبنسلين انت بتعطيه as انجكشن، الولد بكون حرارة وماكل هواء، بعد هذا بعد الانجكشن ثاني سكند ثيرد انجكشن بصير بطيب. خاصة الوقت ذاك الوقت كان البكتيريا is not كانت كتير susceptible، يعني ما عندها resistance، ف it worked very quickly. فكان بالنسبة لهم يعني الريفوليوشن انه شيء مش معقول. ف فهذا كان البنسلين جي. هلا بس البنسلين جي كان فياته مشاكل. اول مشكلة انه was being excreted very rapidly by the kidneys, so it has very less availability in the tissues. You could not give it orally because it was broken down by the acids of the stomach. Broken down obviously by beta lactamases, so works only on gram positives and very few gram negatives. فالسبكترم تبعه ما كان كتير عالي. So هذا خلى الناس يصيروا how can we fix these problems. So the first successful step اللي هو اللي بلشت حركة السيمي synthetic penicillins، and هو removal of a side chain وطلعوا and which left intact the core structure، وطلع لك اللي هو 6 amino penicillin acid اللي هو بلشنا فيه. دخلنا في اللي هو البنسلين في. هلا البنسلين في هو أول واحد that can be given orally. بعدين طلعنا أنواع تانية. البروكين بنسلين is a long acting، فهذا حل لك مشكلة اللي هو being excreted rapidly by the kidney، فحليناها in another type of penicillin. هلا هذا صار resistant for the staphylococcal beta lactamases، فصار عندي رفعت ال شو اسمه السبكترم شوية، فبلشنا نحل المشاكل بهيز. هلا طلع عندك الامبيسيلين والاموكسيسيلين. these are commonly used الآن. يعني من الأشياء كتير أموكلان، إذا سمعتوا عن أموكلان this is very commonly used now. amoxicillin ampicillin they are active against some enterobacteriaceae. وهي حلينا مشكلة السبكترم كمان، رفعنا السبكترم towards the gram negatives. بعدين طلعنا التكراسيلين بيبراسيلين which will work against pseudomonas which they never worked at before. فبلشنا نحل problem 4 as well. So penicillins other than flutoxacillin has its relative and activated by اللي هم staphylococcal beta lactamases. فطلع لنا نوع من انواع البكتيريا of Staph aureus called methicillin resistant Staph aureus. Methicillin is a drug that was used back in the 60s حتى used up to the 70s. It's no longer being used now, but the resistance to it, the beta lactamase that is made against it, makes this bacteria resistant to a lot of penicillins. all beta lactams تقريبا antibiotics will be ما بيشتغلوا عليهم، وحتى كمان انواع من cephalosporins. فهذا كيف صارت الريزيستنس؟ resistance is due to the alteration of in the target. البنسلين بايندينج بروتين كله راح تغيرت شكله كل البكتيريا، فصار البنسلين وانواع البنسلين كلها ما ترتبط على هالبنسلين بايندينج بروتين. فهذا تغيير شكل البنسلين بايندينج بروتين can be transmitted between bacteria and that makes the resistance to this drug, these class of drugs is very bad. عشان هيك مرسى كتير مشهورة، عشان هيك مرسى is a big problem. كمان المرسى because we use a lot of the penicillins. هم أكبر. إذا بتذكروا نرجع للتابل here. Our main uses of these staph and strep was these penicillins and the cell wall drugs. That's a big problem if I can't use the cell wall drugs here. So as penicillins will inhibit transportation, this occurs between the amino group on the end. ذكر حكينا بالمحاضرة الأولى that the chemistry of the drug that it binds to the last two terminal amino acids هم dealanine. That's what penicillin can bind to this drug. إذا أنت بتعمل drug that looks like these two amino acids, it will bind to penicillin binding protein and not let it go. فهذا penicillin binding protein بطل يقدر يشتغل. ف that's how it works basically. So these side chains are brought in together by the penicillin binding protein. انت بتتعرف على هاي الستراكشر by D-ala. So you give a drug that looks like these two amino acids, بقعد هنا وما بخلي هذا البنسلين binding protein to work. This is a more clearer picture. The D-alanine, L or D, these are the red, and you can see between the two, these are the sugar backbone, and these are the amino acids. So you have the alanine here, the two. This is and this is penicillin binding protein. So it finds this, binds the alanine to the next one on the sugar, in this case lysine. تربطهم مع بعض، تكب واحدة من دي ألانين، وبعدين بتروح تنتقل للمرحلة الثانية للجنبيها وللجنبيها. But this is penicillin. Penicillin just looks like the D-alanine, and then it irreversibly binds to the penicillin binding protein. فthat's the structure. هاي دي-ala جنب بعض، لما يكون دي-ala جنب بعض هيك شكلهم. penicillin هيك شكله. This is so that's the mechanism of action. So penicillin, bactericidal or bacteriostatic? Obviously cidal, because you cannot have وخاصة the growing cells, because the cell cannot live without its cell wall. Therefore penicillins are more active drugs on the log phase. حكينا bacterial cell growth. During stationary phase لما يكونوا بالstationary phase خلص they form the layer, فبكون أكثر static عليهم. فخاص إذا كانوا early on in the infection, they are in the log phase, they are more bactericidal and can be more effective during that period. فهي الأنواع penicillin G, the common penicillin and what we use it against. Amoxicillin and ampicillin that we can now use have more activity against the gram negatives, and the respiratory gram negatives, Haemophilus, E. coli, Salmonella, Shigella, but does not work on Pseudomonas. That's what we have ticnicillin and piperacillin for. خاصة هاي كلمة كتير مهمة synergistic with aminoglycosides. راح نحكي أكثر من مرة. Penicillins, beta-lactams and aminoglycosides have a synergistic activity. فهذا كتير تستخدم بالطب خاصة in مثلا in preemptive treatment. مثلا you have somebody, you don't know what is the infection, you don't know, or you know the infection but you don't know what is the resistance pattern. فعادة نعطيهم treatment prophylactically to treat the bacteria. فعادة من الأنواع تستخدم هي بيعطوا penicillin مع aminoglycoside because they have synergism. Synergism يعني 1 زائد 1 تساوي 5 أو 4، مش إنه 1 زائد 1 تساوي 2. So the effect is heightened when you use them together. Piperacillin again similar to ticnicillin. Nafcillin, dicloxacillin, flucloxacillin هذول penicillinase producing. They are against the beta-lactamase اللي هم بالستاف. So these are used especially for staph, anti-staph medications. هلا ننتقل للنوع الثاني من الانتي سيل وول medications واللي هم السيفالوسبورينز. السيفالوسبورينز ولاد عم البنسلينز. They are very close cousins to penicillins. هلا البيتا-لاكتام ring is fused to a 6-membered dihydrothiazine ring. لساتهم بيتا لاكتامز، لساتنا بالبيتا لاكتام، لساتنا the same close to the same mechanism of action. The extra carbon causes alteration in pharmacological behavior لهاي المولكيول، ومنها كمان صاروا يطلعوا أنواع more semi-synthetic mechanism. فزي قصة البنسلين بلسنا عندنا أول جنريشن من السيفالوسبورين كان فيها مشاكل، حليناهم هاي المشاكل وصرنا نزيد ونعدل عليهم. So some alterations work, some cause side effects, some مثلا have other مثلا side effects. So some of them مثلا they can possess methyl tetrazole substituent which has been associated with reduced thrombin. معناته بصير عند بعض الناس معناته بصير عندهم more less coagulation، معناته there's more bleeding in these kind of patients. فهاي هم الأولاد العم. So penicillin next to the cephalosporin. they look very next, يعني it's almost identical. You have the same يعني side chain that might have different things that you can put here, another side chain at the other 3 position, and then you have the beta-lactam ring. In this case is 5-membered here carbon. This is a 6-membered with different. This is the dihydrothiosidine ring in cephalosporins. هلا شو الادفانتجز؟ ان هم they are generally resistant to the penicillinases، خاصة اللي يستخدموا على الستاف. فهذا خلى اللي هم خاصة ال first generation وما بعده are they work against very well against gram positives especially staph. اللي حكيناهم extended spectrum beta lactamases the spectrum is from penicillins to cephalosporins. فصاروا يشتغلوا على cephalosporins and destroy cephalosporins, and that's what makes them a very big problem. Another advantage is that they have a broader spectrum of activity أكثر من penicillin. They have less hypersensitivity. تذكروا حكينا penicillin is a hapten and can cause allergy. ال change in structure بخلي هذا الcephalosporins ما يعملوا allergy بنفس الكمية، فبيعملوا 10%. الواحد بيكون إذا عندك 10 ناس معهم penicillin allergy، واحد من هال10 بيكون كمان sensitive للcephalosporin. فسؤال، هل بتستخدم مع cephalosporin إذا واحد بحكي لك أنا penicillin allergy 9 out of 10 he will be fine, but that 1 out of 10 is enough for you not to use it. Or you can test the skin. تشوف سكين مثلا الأطفال قبل ما يعطوهم مثلا third generation cephalosporin، عادة بحطوا لهم سكين تيست، بستنى نص ساعة. إذا صار في allergy ما بيستخدموه، ما في allergy بروح بيستخدموا cephalosporin، and that can is used يعني very periodically in pediatrics. Cephalosporins are effective against a broad range of organisms, and they are generally well tolerated. And نفس قصة البنسلين، cephalosporins are also products of a mold of genus اسمه cephalosporium، ومنه صار يطلع a lot of synthetic and semi-synthetic semi-synthetic alterations. هلا بعضهم مثلا cefoxytin made by another form من another form of yeast. This is Streptomyces. Among the most important cephalosporin therapeutic uses هم we have now 5 generations of cephalosporins. هاي السلايد عادة أنا بستخدمها عشان أذكر حالي بالأنواع، ولما كنت طالب يعني I had this idea that helped me remember. So مثلا cephalaxin, cefradine, cefaclor، عادة FA فيهم FA. These are usually given orally. Cefuroxime, cefoxetine, so fur, fox, these are the ones that are stable against beta-lactamase. These are the second generation. Cefotaxime, cefotritax, this is the third generations, and have a much higher beta-lactamase stability and very high intrinsic activity. These are يعني life-saving drugs خاصة meningitis and life-threatening infections, especially with gram negatives, and they can cover gram positive as well. Cefopodoxine and cefixine, these are the بيشتغلوا on antibacteria as well. Ceftazidime, these are the ones that work against and cefpiridim بيشتغلوا على Pseudomonas. Ceftaroline, this is work against MRSA. فخلينا نحط هلا هاي السلايد مني، هاي this is a slide from the book. So let's start going through them, and then we will continue إن شاء الله المحاضرة القادمة. So خلينا نطلع على الكلينيكال اكتيفيتي بعدين. هذا هلا عندي حكينا the FA these are the first generation. مش نفس البنسلينز، كان الفيرست جنريشن كتير قوي على الجرام بوزيتيفز، بعدين السكند ثيرد فورث جنريشن زاد الاكتيفيتي على الجرام نيجاتيفز. فالفيرست جنريشن is works in the gram positive staph and strep، طبعا ما بشتغل على المرسى. هلا على السكند جنريشن صرنا نشتغل على الجرام نيجاتيف رودز عن انايروبس. I improved هاي السيفروكسيم الفر and fox. These are the second generations, and these are the ones that have improved their activity on the gram negatives and anaerobes. Third generation صار يشتغل هم هذول سيفتراكسون هم مش تدرسوا فينا وسمعتوا عنه. This is a very great drug that is used حاليا مستخدم for life-threatening infections. بتشتغل على الانتريكس and also on Neisseria gonorrhea and Neisseria meningitis as well. So covers all your bases with this. Ceftazidime هم تبع السودوموناس and works on other gram. هلا fourth generation cefepime بيشتغل على anti-gram negative rods مع that produce the ESBL extended spectrum beta lactamase. So this is the fourth, and your fifth generation is against antimicrobial microorganisms with antimicrobial resistance patterns. Your fourth generation works against the ESBL producers خاصة anti-gram negatives والstaph aureus بس مش MRSA, and the penicillin resistant Streptococcus pneumonia, forms of Streptococcus pneumonia. Some of them are resistant to penicillin. You can use the fourth generation cephalosporin. The fifth generation شو الانتي resistant mechanism بيشتغل ضدها اللي هو المرسا. So ceftaroline is anti-MRSA. This is هلا بدك تصير تجمع what works against anaerobes, what works against gram negatives, what works against gram positives. شو بيشتغل لي على ESBL وشو بيشتغل لي على المرسا وشو بيشتغل لي على vancomycin resistance. So now we have our first ones against ESBL, our first one against MRSA, and then our first against anaerobes. The other beta lactam agents. خلصنا هيك cephalosporins, خلصنا نحكي عن penicillins. So what are their other mechanisms? Or often other agents. There are still other agents that still have the beta lactam ring. اللي أهم منهم الأنواع اللي هم الكاربابينمز, الإرتابينم, الميرابينم, إيمابينم. These have broad spectrum activity, بيشتغلوا على gram positives و gram negatives والanaerobes. عادة يستخدموا كمان for the ESBL producers, بعض أنواع ال ESBL producers as well. So imipenem is inactivated by the kidney, and you have to add cilastatin with it to reduce this dehydropeptidase, which reduces its activity. Imipenem is prescribed with cilastatin زي ما حكينا to improve its activity, and it is not it's not inactivated by beta lactamases. However, carbapinamases هاي المشكلة جديدة. صار نحكي بالبيتا لاكتاميز, بعدين اكسدنا سبكترم بيتا لاكتاميز, بعدين صار عندنا كاربابينامينيز. هذا هو المشكلة رقم 3 بالجرام نيجاتيفز, خاصة produce كليبسيلا and degrades these carbapinamases. تذكروا التيبل وين راحت الصورة؟ حكيناها بأول محاضرة. هاي حكينا صرنا نحكي carbapinams, how they are one of the last defenses that we use, and how we have an uptick in E. coli and Klebsiella هنا, and that's the carbapenem that we're talking about here. So yes, other carbapenems, في عندنا ertapenem and imipenem. You don't have to use cilastatin, and they are used against pathogens that are complicated infections, and these are usually given in the hospital. يعني under the care of physicians and people that are admitted to the hospital. ما بنستخدمهم خارج المستشفى. So this is something that we can use against the ESBL producers. Monobactam, astrionam, is a monocyclic compound that works. Is restricted. Monobactam هذا استخدام على gram negative bacteria. Is the most useful monobactam هو astrionam. بيستخدموا على الجرام negative rods هم Enterobacteriaceae and Pseudomonas. Clavam اللي هو clavulanic acid. Clavulanic acid does not have antibacterial activity really, but it inhibits beta lactamase. فإذا بستخدم الكلافام مع drug مثلا amoxicillin, أنا في عندي amoxicillin resistance, لأنه في بيتا لاكتاميز, فهذا بوقف البيتا لاكتاميز بصير amoxicillin يشتغل. تقدر تستخدم مع amoxicillin, مع تكراسيلين, مع ampicillin, كثير من أنواع ممكن تستخدم معهم الكلافام which improves their activity by reducing or eliminating the beta lactamase enzyme. هذا بسموه مثلا إذا بتسمعوا عن الأوجمنتين أو أموكسيكلاف، هدول هم بيستخدموا كلافيورونيك أسيد مع أموكسيسيلين مثلا. بس آخر إشي خلينا نحكي هنا اللي هو الجلايكوبيبتايدز اللي هم الفانكومايسين and teicoplanin. These are large molecules that are unable to penetrate the cells. فمعناتهم they can go through the outer membrane, but بالنسبة إلى الجرام positives they can penetrate بدخلوا بالبعضوا على هالجرام على هالglycan layer, and this is where actually they work. So that's why they can only be used on gram positives. So now I have acidurinam that can be only used on gram negatives, and then glycopeptides that can only be used on gram positive. The chief importance is gram positives وأهم إشي هم المرسى. فناو this is my مع سفتارولين. هلا صار عندي الدواء الثاني اللي هو أو أهم الدواء اللي هو glycopeptides. Actually I use glycopeptides before I even think about using ceftaroline. So vancomycin اللي هو your go-to drug عادة for MRSA, and enterococci, staphylococci including MRSA and that exhibit resistant to reduced sensitivity to glycopeptides are being reported. فصار عندنا كمان versa vancomycin resistant enterococci, vancomycin resistant staph aureus. So now this is another problem. معناته بدنا يكون عندنا drugs that we can use against these vancomycin glycopeptide resistant pathogens. Let us stop here for lecture 1. I will continue lecture 2 by نخلص cell wall drugs وندخل على anti-protein drugs.